அது பார்ப்போமா அதில் மதிப்பெண் இதுக்குள்ளேயே 3 வருடங்கள் சொல்றது என்ன பண்ணி போடணும் பக்கத்தில் இங்கே வருங்கள விண்டோ நினைக்கிறேன் விண்டோல தான் இப்போ நம்ம போட்டுருக்கணும் ரெண்டே ஸ்க்ரீன்ல போட்டுருக்கோம் ரெண்டே ஸ்க்ரீன் போட்டு அவங்களுக்கு ஃபுல்லாக வந்துருக்கோம் இதுல இந்த ஏதோ ஒரு ஆப்ஷன் ஒன்று இருக்குது போனோம்னா அந்த சார் தெரியல இல்லையா அதில் போய் ஃபுல் ஸ்க்ரீனா வேற ஏதாவது மாறிடுச்சுன்னு இது இது மற்றவங்க எல்லாம் கீழே சைடில் வரும் அது மாதிரி இப்போ கொடுத்து பார்க்கணும் ஐயோ இருக்கிறது எது மாதிரி தெரியும் அது பேருக்கு மேடம் சொல்லுங்க அப்படிங்களா ஒரே நிமிஷம் மேடம் ஒரே நிமிஷம் இருங்க அது இங்க ஓகே ஓகே அது நீங்க அது பேசும்போது நீங்க ஆன் பண்ணிருங்க இது இங்க ஹைடிங் ஆகிட்டு இருந்துச்சு அதனாலதான்
Sir, shall we start the program, sir?
Kannan, sir.
Okay, sir.
Yeah, sir. Good afternoon. Okay, sir. Start the program, sir. No problem.
Okay, sir. Okay, thank you, sir.
Okay, sir. Okay. Can I mute the microphone, sir? Or?
No, sir. No, sir.
Okay. Okay. Thank you, sir.
Sir.
Karan?
Sir, proceed பண்ணலாமா sir?
Yes, proceed.
Thanks, sir.
Good afternoon, all. I invite you all for the third national webinar conducted by Department of Microbiology, KR College of Arts and Science. To begin the webinar, I'd like to request our HOD, Mr. Kannan sir, to welcome all. Over to you, sir. Kannan sir.
Okay, madam. Thank you, madam. Thank you. Hello? Is it okay, madam? Okay. To respect your resource person of today's national webinar, Dr. P. Tangavel sir. The respected principal of our college, Dr. S. Respected head of the department of various discipline, faculties of our college, participants of the institution. Good afternoon to all. It's my immense pleasure to give a cheerful welcome to our resource person webinar, Dr. P. Tangavel, Assistant Professor, Department of Environmental Science, School of Life Sciences, Periyar University, Salem, has accepted our invitation. Busy schedule. Bio-Geotechnologies for remediation of metal contaminated soil. On behalf of our management, principal, faculties, we wholeheartedly welcome you, sir. Welcome, sir.
Thank you, sir. Thank you.
Like, okay, sir. Okay, thank you, sir. My desire is to extend a gracious and inclusive welcome to our respected principal, Dr. S. Madhivanan, sir. Our principal is an example of professional excellence. He set high standards for others to strive forward. His outstanding contribution help us and our student to stand above them the top. His motivation and determination lead our students to higher performance. He is a key to our success. On behalf of our departments and faculty members, we proudly welcome you, sir. Welcome, sir.
Thank you, Ganan.
Next, I extend heartiest welcome to faculty members, scientists, and research scholars from other institutions, head of the departments, faculty members, and student friends from our college. I hope that today's national webinar will be exuberant, instrumental, and favorable for your future studies and research. Once again, I extend a warm and cordial welcome to one and all. Thank you. Thank you, sir. Over to Sophia, madam.
Thank you, sir. I request our beloved principal, sir, Dr. S. Madhivanan, sir, to felicitate us.
Thank you, madam. The most respected resource person, Dr. P. Thangavel, faculty members, scientists from participating institutions, HODs and faculty members of our college, alumni of Department of Microbiology of Care College of Arts and Science, scholars, and my dear students. Good afternoon, you all. It is my privilege to felicitate and wish the participants of this national webinar on the biotechnologies for remediation of metal contaminated soils organized by the Department of Microbiology of our college. This wonderful program is arranged particularly during this pandemic period to discuss about remediation, to explore biotechnologies implemented in remediating metal contaminated soils by our resource person Dr. P. Thangavel, Assistant Professor, Department of Environmental Science, School of Life Sciences, Periyar University, Salem. The resource person, Dr. P. Thangavel, having 24 years of research and teaching experience, and his field of interest is soil ecology, Phytoremediation, environmental impact assessment, and biofuels. I am sure that the experience and expertise knowledge of our resource person will be much useful to the participants to understand the concepts of today's discussion. I take this opportunity to welcome and thank the resource person Dr. P. Thangavel, who has accepted our invitation during his busy schedule and also the period of pandemic to share his knowledge and research experience in the field of remediation in today's webinar. On behalf of management and faculty members of our college, I welcome all the active participants and also I wish for the grand success of this webinar. During this scientific session, our resource person will highlight the various current technologies and its application available in the field of remediation. I hope that this webinar will be highly informative and interactive to share the ideas of scientific thoughts among you. Finally, it is my duty to appreciate and to congratulate the head and faculty members of Microbiology Department of our college for organizing such a wonderful international webinar for the benefit of students, scholars, and faculty members of life science discipline from different institutions across India. Let me conclude by wishing that each one of you will carry back from here some wonderful scientific thoughts that will help you to flourish in the field of science. Good luck for you all. Wish you all the best. And once again, I thank you one and all.
Thank you, sir. I request Dr. Sanjeev Kumar, Assistant Professor of Microbiology, to introduce the resource person for this webinar.
Thank you, madam. It's my great privilege to introduce our today's chief guest, Dr. P. Thangavel, sir, as an Assistant Professor, Department of Environmental Science, Periyar University, Salem. He has completed his UG/PG degree in zoology from Bharatiyar University and MPhil degree in zoology interdisciplinary environmental science from Bardiyar University and also completed his PhD degree in environmental science from the same university during 1996 to 2002. At that time, he has elected in the position of Senior Research Fellow and Research Associate, etc. He has started his career as a guest lecturer in 11 to 13 and then switched over to the assistant professor from 2013 onwards. He is a well-known person in the field of environmental studies, soil ecology, phytoremediation, biofuel, and fortification, etc. He has visited various countries like USA, Canada, China, Thailand, South Korea, and Slovakia, etc. He got various awards and fellowships like Senior Research Fellow and Research Associateship from CSIR New Delhi, Foreign Travel Grant from CSIR DST, and Best Scientist Award in the Environmental Science from Pearl Foundation, Tamil Nadu. He has elected as a resource person and participated and presented his research findings in various conferences. He has published more than 50 research articles in the reputed peer-reviewed journals with high cumulative impact factors. He has also published various books and book chapters, and he has successfully completed various funded projects nearby the cost of one crore issued by DST, SERP, MOEF, etc. These are some words about our eminent resource person. Thank you, sir, being a part of our webinar today. Thank you. Thank you once again, sir.
Thank you, sir.
Sir, it's over to you, sir. You can now start the session, sir.
Okay, madam. Is it audible, madam?
Sir, it's audible, sir.
Okay, can I show the slide for full view?
Yes, sir. It's maximize it, sir.
Can I start, madam?
Yes, sir. Yes, sir. You can start, sir.
Okay. Is it audible?
Yes, sir. It's clear, sir.
Okay. Okay, madam. Good afternoon for all. And first of all, I would like to thank respected principal and Dr. Kannan, head of the department microbiology, and other faculty members, Dr. Sherilyn and Dr. Subbulakshmi, for giving me an opportunity to share my views in your esteemed platform and in my views on Bio-Geotechnologies for remediation of metal contaminated soils. And in the next 1 hour talk, I would like to mention very briefly about the different categories of phytoremediation and how we used it for both the rhizosphere and plant-based remedial mechanisms, the two types of remedial mechanisms, and I would like to highlight exclusively for the phytoextraction. If time permits, I would like to explain about the phytomining and agromining, and we will show some of the successful phytoremediation trial experiments throughout the world to utilize it for the remediation of metal contaminated soils. Why we paid more attention about the soil functions, soil diversity, because the soil deliver different ecosystem functions like nutrient cycling, climate regulation, carbon sequestration, cultural heritage, and it is a source of pharmaceutical and genetical resources. And also it is a center part for the food security as well as the water security, and it's the heart of both the things. And these two things, food security and water security, is most important component for our sustainable development goals recently proposed by the UNDP. In 17 sustainable development goals, and the food security and water security are also one of the things. And here in the recent report by Institute for Advanced Sustainability Studies in Germany, and they said we already knew very well about the carbon sequestration is exclusively for trees. But the scenario is not like that. In Africa, the carbon sequestration is in soil also. Like in Europe, see that entering the carbon in soil more than in trees. But the scenario in Africa is entirely different. So soil also play a major role for carbon sequestration. And also today's scenario is the soil has been deteriorated or polluted in several aspects like pollution, desertification, deforestation, degradation, soil erosion, and soil urbanization or soil sealing, and so many factors. And why we need to pay more attention about that the soil, extent of soil global pollution, because we need to pay more attention not only about the soil pollution, not only in India, even for the global perspectives. In global estimation of soil pollution is at present 22 million hectares at present. And according to the Chinese Environmental Protection Ministry, and they have to publish 16% of all the Chinese soils, including 19% of agricultural soils has been already contaminated. And according to the European Environmental Association, economic area is approximately 3 million potentially polluted sites has been identified. And in USA, 1,300 polluted or contaminated sites has been identified. And Australia, more than 80,000 sites. And there is a urgent need, but such kind of a scenario or soil pollution atlas mapping is not available in India and other developing countries. This is one of the very pathetic situation. So that's why the Global Soil Partnership GSP program of FAO, they have to urge or they have to pay more attention about the global soil pollution aspects and to implement the global assessment of for soil pollution in that scenario. And in addition to that, several factors, and recently our country, India, hosts the UNCCD conference in New Delhi, and soil degradation or land degradation, because 12 million hectares of produced lands has been already degraded, and that will equal for 20 million tons of grain productivity. And 1/4 of the global land has been already degraded for the past 2 decades. So this is that's one of the important aspects or important constraint factor for the soil quality. And already 96.4 million hectares of the land has been already degraded, and we need to restore such kind of the degraded land based on that several aspects. So that's why India's new target is 26 million hectare of the lands we need to restore from the soil degradation aspects. And this review, this pictorial representation is mentioned about how many sites are potentially contaminated and how many are contaminated and how many are remediated according to the European Union based on the Panos et al. Review. And they have to pay more attention about the remediation technologies. And this pictorial representation is mentioned about the soil pollution prevention and control Axel plan in 2016 by the State Council of China, and to identify the metal contaminated sites, and according to the 13th 5-year plan. And already based on that program, and they have already implemented more than 100 remediation projects for the past 10 years. And according to the world global scenario, and 10 most top polluted cities in the world, Sukinta in Odisha and Vapi in Gujarat also occupied the top 10 position because of lot of chromite mines and mercury levels contaminated in that area. And why we need to pay more attention about the soil pollution? Because in the recent review about the Who et al. By Nature recently last week, and so many among the 17 Sustainable Development Goals, so many goals are to that SDG about the soil pollution, including the SDG 1, no poverty, zero hunger, good health, clean water, climate action, life on land, and so many. Nearly 8 SDGs are directly linked with the soil pollution scenario among the 17 Sustainable Development Goals. And as you are already well known about the sources of soil pollution, I would like slightly mention about different sources like fertilizers. Nowadays, for the modern agricultural practices, we input so many inorganic fertilizers like NPK into that to increase the agricultural productivity to ensure our food security. And especially for the phosphate fertilizer, it is one of the major constraint for the cadmium pollution. That's the major constraint for all the human being and other things, Ita disease and others in human being. And manures, irrigation purposes, sludge applications, and industrial applications like waste incineration, or fossil fuel applications, fossil fuel coal-fired power plants, and they have to emitted and aerial depositions and running into that deposited agricultural soil and irrigated wastewater from that industrial resources. And mining operations are AMD, acid mine drainage, and these are all also even for mine extraction or smelting. And these are all enters into that agricultural soil or other soil, and that will have contaminated with the soil environment. It is one of the major component. And why we need to pay more attention? This we have already seen. Even though the remedial technology, 3 types of remedial technologies are there: physical technology, chemical technologies, and biological technologies. And both the physical and chemical technologies, they have using destructive for the soil physicochemical properties, and that's why we need to utilize it for the biological methods or bioremediation and phytoremediation. Bioremediation in general we use it for microbes exclusively. For phytoremediation we use it for plant to remove the contaminants, either organic or inorganic, and without destruction, without affect any soil quality parameters. That is the most important. Heavy metals rather than the other organic pollutant, because heavy metal has a more persistence time. Once it's removal of heavy metals from the soil, it's very difficult. For example, resistance time is more than 200 years in that soil. So, in general, we have to divide into 2 types of technologies among the physical, chemical, and biological. One is isolation and containment technology, and another one is removal technology. Containment technology to arrest or to contain that metal pollution sources by using several physicochemical methods, and removal technology by using heavy metals from the substrate into the above ground parts, and later we have converted into the potentially utilized applications by using pyrolysis and other things like phytomining and agromining aspects. And yes, I have already mentioned in that the initial slides, previous slides, like both the rhizosphere-based mechanism and the plant-based mechanisms are there for the remedial mechanism. And what are the rhizosphere management? Due to the time constraint, I am not going to in-depth about the each steps. So I would like to highlight significant points in these rhizosphere management mechanisms. And the success behind that phytoremediation technology is to bioavailability of heavy metals and the type of the plant species. So we need to amend different chelators to enhance the bioavailability or metal mobility or metal availability in the substrate, like EDTA, HDTA, and NTA citrate and other synthetic zeolites. And soil fertilization, some kind of NPK, they have application to that soil, even in contaminated soil, and to enhance the biomass of that particular plant and metal accumulation in that soot. And phytosiderophore formation in Poaceae family, especially mugenic and avinic acid, and that will increase the root uptake of iron, and alteration of the physiochemical characteristics in that rhizodeposition soil and root portion, rhizosphere portion, and root exudate secretion, for example, organic acids, that will also increase the mobility of heavy metals, and alteration of the rhizosphere pH, that's one of the strategy. Manipulation of the sorption sites, and we need to increase the phosphate to the soil and to extract arsenic. That kind of some interactions between the phosphorus and arsenic, because they have the same similarity of phosphorus and arsenic uptake. They have to utilize the same transporter. Similar to that of selenium and sulfate, they utilize the same transporter for metal uptake and other things. And the rhizosphere mediated metal remedial mechanisms. And the bioremediation, and I'm not going to in-depth for bioremediation also, and for that different options are there. Biosorption and bacteria, they have to interact with metal or metalloids. Metalloids means oxidation states of metal like arsenic, selenium, mercury, and these things are metalloids. And the bacteria can interact with heavy metals or metalloid directly and accumulating into that cell. That is called biosorption. And for the biosynthesis or biodegradation, they have to reduce or oxidize the metalloids directly, and oxidize the metalloids and synthesis or degraded. That's the biodegradation or biosynthesis. Metal containing organic compound especially, and bio-leaching, sulfur oxidizing bacteria can release acids and dissolves the metal containing compounds from the metal ores. And bioprecipitations and sulfate reducing bacteria can precipitate metals by formation of the low mobility sulfates. That's the bio precipitation aspects. And for that bioaccumulation and accumulate metals in the intracellular space of microorganisms, cellular spaces, bioassimilation and assimilate metals via ion assimilation pathways by using the siderophores. And these are all the different kinds of microbial bioremediation methods are utilized normally to remove the contaminants. And several microbes are used, even Micrococcus, Pseudomonas aeruginosus, Micrococcus, several species, Klebsiella pneumonia. And what are the major pros and cons of these things recently reviewed by Daliva et al. 2002? And what are the pros and cons? The constraint is they have, for such kind of the microbial remediation or bioremediation, we used it to provide suitable conditions for their growth in sometime in laborious. And in addition to that bacteria, arbosolar mycorrhizal fungi, and that is the root fungi associated with the rhizosphere soil, and that AM fungi also involved for the remediation purpose in both the stabilization aspects as well as extraction. And we will see the stabilization is to arrest the metals up to the substrate, and for the remedy extraction is to mycorrhizae is associated with to enhance the metal mobility into the above ground level. And this is the review reported in the Biotechnological Advances by Rajkumar et al. And how the microbial, the arbosolar mycorrhizae and other rhizosphere bacteria in both the stabilization aspects as well as phytoextraction aspects. Stabilization means to arrest the metals mobility up to the substrate level, and for the extraction is to enhance the metal mobility to the above ground parts by various aspects like metal mobilizing chelators associated with translocation and immobilizing the metabolites, and so many mechanisms, such are rhizosphere based remedial mechanisms up to 2010, and now it's another 1 decade later before, and so many research articles has been published in Earth Science Reviews and so many environmental journals. And then coming to the plant-based remedial mechanism and how the plants utilized for the remediation of metal contaminated soils. The strategy has been implemented first in nickel accumulation by Alison Bertoloni in the year of 1948, and the concept was implemented by Robert Brooks in New Zealand from Massey University in 1977. Later, this strategy has been implemented by nuclear disaster at Chernobyl by Ukraine in the year of 1986 by Phytotech company by M. Lassard. And later, and that can be utilized into that Iowa City tree farms or landfills. So many categories are available in that phytoremediation strategies: phytoextraction, accumulation, filtration, stabilization, volatilization, rhizodegradation, so many things for organic pollutant as well as for the inorganic metal pollutions. And due to the time constraint, I am not going to in-depth about things. And what are the major key categories of phytoremediation? Here we would like to see phytoextraction is to remove the contaminants from the soil or substrate into the above ground plant pots, especially in the bacular portion. And phytodegradation is plants and associated microbes. They have to degrade the toxic into non-toxic substances by using several microbes. Especially these methods, it is used for only for organic contaminant. And rhizofiltration, and they have to plant roots utilized for the absorbs metal pollutant or other pollutant from wastewater. Blastofiltration is seedlings absorbs metals from the wastewater. Then the blastofiltration strategy normally used for the bioassay studies, whether such plant can be able to tolerate for the particular conditions or site condition or not. So for the laboratory assay or other bioassays, we have to utilize the blastofiltration study. Phytostabilization is to arrest the metals up to the substrate level by using different organic amendments or synthetic amendments or immobilization strategy, and mainly it is used for the mine site reclamation purposes. And phytovolatilization is especially for the metalloid contaminants, arsenic, selenium, and mercury, and volatilization of pollutant into the atmosphere via plants. And this we would like to highlight only for phytoextraction strategy and how the plants utilize it for that strategy, and we would like to see here. And as I have already mentioned, phytoextraction is the contaminant or metals from the accumulated in the above ground plant parts like shoots, especially in the vacuolar portion of leaves. And due to different strategies of phytoextraction when these has been utilized nowadays have been deployed. Natural hyperaccumulation and to enhance the bioavailability without addition of any chelating agents or any additional substrate here. That's the natural hyperaccumulation. The plant can able to accumulate more amount of the heavy metal in the above ground parts without adding any chelating agent. But induced hyperaccumulation, we need to increase the metal mobility and transpiration rate by adding some of the chelating agents like EDTA, NTA, synthetic zeolites, and some other chelating agents. Increase the bioavailability of heavy metals in the soil, and also that will indirectly increase the transpiration rate and increase the uptake of heavy metals in the above ground plant parts. And in general, the bioavailability of heavy metals can be classified into three different types: readily bioavailable, moderately bioavailable, and wholly bioavailable. As I have already mentioned in the previous slides, why we need to pay more attention about heavy metals, because lead is the longer persistence times, 200 years. Once it's accumulated onto the soil, the removal is very difficult. And readily bioavailable, cadmium, nickel, arsenic, selenium, copper, so many metals. So the successful of that phytoextraction strategies entirely depends upon the type of the contaminant and the type of the plants we chosen. And in general, the metalliferous habitat, metal contaminated sites can be classified into three types according to the Baker et al. Professor Alan Baker is a pioneer man in the field of phytoremediation. Primary sites, it's a metal ore or outcropping, naturally metal outcropping soils, mostly in that without any anthropogenic impact, Central Europe, Alps, Balkan region. Secondary sites, by using metal contamination by mining process, ion deposition of soil, and slug heaps, other soil alteration. Approximately 1% of global surface, soil surface or land surface has been affected by the mining activities. Tertiary metalliferous soil mostly for the industrial or anthropogenic contamination. Several anthropogenic activities like industrial, agricultural, sludge application, municipal wastewater, transportation, urbanizations like. And then again, they have to classify into primary and secondary sites depending upon the physicochemical properties of that soil or that the geogenic in nature. Serpentine soil or ultramorphic, mostly in that nickel enriched soil, and calamine soil enriched mainly with zinc and lead and cadmium, mostly located in South Asian region and some Europe region. And the serpentine is mostly in New Caledonia, Cuba, or Turkey. Soil rich with cobalt or copper in Central Africa, especially in the Dominican Republic, Congo, and Zambia. And seleniferous soil, selenium contamination in Australia, Southern Asia, Ireland, US also, and in India, Punjab. In some parts of the Punjab, it's highly contained with the seleniferous soil. And similarly for arsenic also in Bangladesh and Calcutta, lowland areas contaminated with arsenic in rice field and also others. And in general, the metal, any metal accumulating plants or metallophytes, ecologically they have to classify according to the Antonovics by metallophytes. Any plant can occur metal enriched soil. That's a metallophyte. Elective, more vigor or more growth on metal contaminated soils. That's the elective pseudometallophytes. Indifferent pseudometallophytes, equally they have to sporadically distributed really appeared in that areas. And one of the key criteria for that strategy is plant metal tolerance. The plants can able to tolerate heavy metals in that's one of the important strategy. So according to the Baker, and they have to classify it into excluder plant, indicator plant, and accumulator plant. Excluder plant, they didn't allow any metal accumulation in that plant portion. Indicator, they allowed, but without any toxic symptoms. And in accumulator, they accumulate more metal without any toxic symptoms. And later, linen dots and Van der Ende dot linen dots, they have to redefine. Are you able to audible?
Yes, sir.
Are you able to audible,
Yes,
madam?
sir.
Okay. And
Yes, sir. Yes, sir.
linen dots, they have to redefine for the classification Heavy metal sensitive plants, it is more sensitive to heavy metals. That plant cannot able to survive into that contaminated soils. And heavy metal resistant but excluder plant, they have to resist the heavy metal toxicity in the contaminated sites but exclude the heavy metal accumulation. They can't able to uptake the heavy metal contamination. And heavy metal tolerant and hyperaccumulator, that plant can able to accumulate more heavy metals in the root portion rather than the above ground parts. But heavy metal hyperaccumulator or hyper tolerant, this is the ideal plant for phytoextraction strategy because they have to able to accumulate more metals in the above ground parts, especially in the vascular portion. And so many metal transporters are used. HMA3, HMA4 and NRAM and CAX transporters, depending upon the type of the heavy metals and depending upon the type of the plant parts, they have to utilize a different type of the metal transporter. This is the intrusion or exclusion and across the cell membrane and inside the cytosol, and they can enter into the vascular sequestration. And these are all the different transporters, and they have to utilize it for different type of the plants. In sensitive plants, all the metal ions are enters into the cytoplasm, but in that hyperaccumulator plants, all the heavy metal ions binded with the thiolates and enters into that vascular sequestration. So that's why it's more tolerance rather than these sensitive plants. And another hyperaccumulator means you knew about the hypertension, high BP, like that higher accumulation of heavy metals in that shoot biomass, like 1% of dry weight. And in addition to that higher accumulation, different threshold values has been proposed by Vander in the year of 2013 for cadmium, selenium 100 mg per kg, cobalt, copper 300, lead, arsenic 1000, zinc in earlier 10,000, now they redefined it as 3000 mg per kg, manganese 10,000 mg per kg of heavy metals that can be accumulated. But in nowadays, so many research articles has been published in open access journals or some sporadically non-SCI journals like that. So many plants, even edible plants also can be utilized for hyperaccumulator plants. But in real scenario, such kind of the plants, even sorghum or allium, amaranthus, and these kind of plants cannot be able to grow in a real mine contaminated soil or highly polluted soils. So any plant that can grow into a natural contaminated soil without adding any alternative or substances, that's called the real hyperaccumulator heroes. So biological criteria, we utilized several criteria, enrichment factor, and even that in recent scenario, in earlier greater than 1, it is called enrichment factor, that's the hyperaccumulator. But now in the recent article by Anthony Addis et al., and they have to redefine it a different enrichment based on that very severe or no enrichment like that. Similar to that of the translocation factor, translocation indices. If that excluder less than 1 and accumulator is greater than 1, hyperaccumulator is greater than 10. Either if it is exclude plant, we can utilize phytostabilization. And hyperaccumulator, we utilize such plant for phytoextraction studies. And as I have already mentioned, metal tolerance is one of the key criteria. So many other criteria also we utilized before to choose the plant for phytoextraction strategy. And that hyperaccumulator is ability to grow outside of the area of collection. And wide geographical area plant like Prosopis juliflora, Velicurvel solluva. And Prosopis juliflora can be used for biofuel and as well as for the remediation purpose. Profuse root system with fastest growth, and the penetration root growth is more in-depth. Short rotation annual crop, metallophilic root response means that plant when the root can be able to live only for metal contaminated soils rather than the controlled soils. And so many rhizobox experiments has been already proved by our former Chinese research group and US research groups. And that metallophilic root response is one of the key criteria. And metal loving plants and high transport, high transpiration rate, and the ability to accumulate multiple metals and several metals. Some plants can be able to accumulate cadmium, zinc, lead also. So multiple metal accumulation capacity and economic interest. But the one important thing is to avoid any minimal food chain transfer possibilities. So that's why we normally we can't use any edible plants for that practices. And even though The global geographical distribution of hyperaccumulators so far is 721 hyperaccumulator database so far identified by Professor Vander et al. And Reeves et al. They have to publish the recent report from wide geographical temperate to tropical environment, and among 75% are nickel hyperaccumulators, nearly 450, more than 450 species, even from Cuba, Southeast Asia, Brazil, Southern Europe, and even zinc and lead from Northern Europe, cobalt and copper, South Central Africa, as I have already mentioned, Congo. And these are in parentheses, these are all the number of hyperaccumulators. So far arsenic, only 5 hyperaccumulator was reported. Cadmium and thallium, only 2 hyperaccumulator was reported. And this phylogenetic tree was published by Kramer in 2010. And this is one of the Arabis paniculata. That plant one is able to accumulate both cadmium and zinc, was identified in our former Chinese research group by Professor Chu Rongliang in Sun Yat-sen University. And I was associated with Professor Chu's lab for more than a year. And these are all some of the example about the hyperaccumulator plant. What are the hyperaccumulator plant? Name Alizum murale for nickel, Astrocalus bisulcatus for selenium, Teriswitata for arsenic, Brassica gensea for cadmium, Thalaspi for cadmium and zinc, Thalaspi alpris zinc. And I have already mentioned about some plants can be able to accumulate both metal. This is the best example, Thalaspi or Noxia cerulescens. It can be able to accumulate both cadmium and zinc, but the same species, Thalaspi alpri, but it can be able to accumulate only zinc. And Hovaniastrum humbly, it's a cobalt hyperaccumulator. And these are all some of the plant recently published article, cobalt and hyperaccumulator from different geographical region in Central Dominic Republic Congo, DR Congo, like Bekia codi, Alizum murale, and hibiscus Hovaniastrum roberti, and so many plant species. And another one important is Rhinoraria bengalensis. This one, Rhinoraria bengalensis, best high accumulator. And this is the species richness and endemic system, the hotspots of hyperaccumulators. Any hotspots, we have to define the 2 important criteria, endemism and the species richness. Based on these 2 key criteria, we have to fix the hotspots of hyperaccumulator. Mostly in New Caledonia, more than 1,200 species nearly they have been identified. But the database, so far only 700 species has been recorded. The different geographical environmental ecosystems, they have to accumulated in that sporadically distributed, even in ultramorphic island, subalpine shrub and mature mixed casuarina forest, mixed forest, mountain clove forest like that. And some of the examples about nickel hyperaccumulator, Philanthus species, Rhinoraria species, Rhinoraria bengalensis, as I already mentioned, from Sabah, Malaysia, and Philanthus species, one of the important, and recent one, Actiphilia ellenbeckeri, and it's one of the recently identified nickel hyperaccumulator from Sabah, Malaysia. And to honor Professor Alan Becker, and as I have already mentioned, he is the pioneer man for father of phytoremediation. And to honor that person and that plant when they can be named as Actiphilia ellenbeckeri, belongs to the family Phyllantheraceae, was identified by Van Teren in the year of 2016. And so many species has been identified, especially in Philanthus balgui, and these phylogenetic tree, especially for selenium accumulation, Professor White. And these are all identified hyperaccumulator, indigenous hyperaccumulator. By Chinese research group, our former research group, Professor Chu Rongliang, in Arabis paniculata, Picris divaricata, and Potentiella griffithii. These 3 plant species we have identified in our research group, and Sedum alfredi even is some other research group from China. And the key criteria is to vacuolar sequestration. More than 80% of the heavy metals, they have been accumulated into that vacuolar portion that can be proved by our indigenous hyperaccumulator Potentiella griffithii, and that has been published in high impact factor journal, hazardous material, 9.3 impact factor. And they have to utilize it for intercropping also, agromining, and cultivated for such kind of sedum, Plumbus inquisicola. This is for used for the mine contamination sites by our colleague Hu Bingji. in Nanyang Technological Sun Yat-sen University, and phytoextraction of cadmium by Sedum plumbis in Korea with the co-cropping with maize in Zhejiang province, and some of the eucalyptus plant manganese hyperaccumulator from China, and the lantana camera unichedi, and this is one of the potential notorious weed belongs to Verbenaceae family, and that can be also utilized as cadmium hyperaccumulator. And the Prosopis juliflora and Velicervel, as I have already mentioned, the dual benefit plant, and that can be utilized for remediation purpose. And in myself and our research group from Bharatiyar University has been identified in the ability of metal accumulation in 2000, in the year of 2000, 2 decades before, and published in the IFRO World Congress. And so many dual benefit plants can be listed for including both organic and inorganic remediation on both organic and inorganic contaminants, miscanthus species, Matuka, Prosopis juliflora, Acacia, Rhinus, Ricinus, Camellia, so many species. So we need to conserve such kind of the species in hyperaccumulator garden, like in in situ conservation by Professor Van der Ende, and is now in France. And this strategy can be used for biofortification application also. And the essential to accumulation of essential elements in the grains and some other above ground plant parts, edible plant parts, and to nullify the nutrient deficiency symptoms. That's the biofortified aspects. And that can be utilized several transporters to increase the zinc accumulation in Arabidopsis gallery. And this is the potential way for biofortification. So far, different hyperaccumulator databases are there. Phytorem database operated by Environment Canada, metals operated by Professor Alan Baker by University of Sheffield. Now he's in University of Melbourne, Australia, and International Serpentine Ecological Society. I am one of the member of this society, and they have to maintain the global metalophyte database operated by Van der Ende. Center for Mainland Rehabilitation, now University of Newcastle, operated by Professor Ravi Naidu and group and his colleagues. And several potential biotechnological strategies also used for these remediation purposes. And various, in case if we utilize it or to enhance the uptake or manipulating the metal transporter genes, influx or efflux, to increase the uptake of heavy metals in the above ground part, or enhance the metal or metalloid accumulation in that ligand production or chelacing aspects inside the vacuole, and volatilization, conversion of metals and metalloid into less toxic by volatilization aspect, several gene editing technologies. And here they used the transgenic approaches, omics approaches, genomic, transcriptomic, proteomics, metabolomics, or phenomics. And we have to identify the particular candidate genes by using different strategies like transgenic approach, gene stacking, pathway engineering, or the modern genome editing technique. Nowadays, we use CRISPR-Cas or genome editing techniques for various human health issues and others. So we utilize it for such kind of the omics approaches or transgenic strategies, and to develop a suitable plants, transgenic plants for hyperaccumulator, engineered hyperaccumulator plant for metal decontamination programs. So many research is going on in Arabidopsis thaliana plant, and the Thelopsis cerulescens is one of the hyperaccumulator because they shares both the DNA identity. Codon is more or less similar, 88.5%. DNA identity is similar in both the things, and so many, as per the bibliometric analysis, so many phytoextraction or remediation technology studies, even though different technologies, phytoextraction, rhizofiltration, stabilization, and mostly more than 50% of the technologies belongs to the phytoextraction, and the remaining for rhizofiltration or the aquatic thing, and so many others, phytostabilization and other things. So research is going on for the past 2 decades. This is the how in abroad they have to some consultancy or like that phytotech companies, they have to utilize some standard protocol for remediation of metal contaminated sites. This is the standard protocol for management of contaminated sites. That the main objective or main goal or the fundamental take-home message is we need to get some economic benefit opportunities for farmers or some stakeholder. That's the one of the main important criteria. And they have to get the economic opportunities by using biofortification, bioenergy, timber, or biochar options. So based on these different options, we can get economic benefits to the stakeholders. And these are all, even for remediation is one of the goal. And at the same time, we can get some economic benefits also for that particular project. And the cost benefit also, it is a very cost-wise, it's a feasible one. It's an eco-friendly, feasible, cost-effective technology, and without addition of any adverse effects on soil, physicochemical or biological characteristics, and aesthetically pleasing, and it's used for wide range of contaminant, minimal environmental disturbance of limitations, because it's a time consumable. We need to identify the suitable land. We need to identify suitable indigenous hyperaccumulator plan, dual benefit plan, metal tolerance characteristics, and minimizing the dispersal of seeds and the food chain transfer limitations. So many things we need to pay more attention before implementing such kind of these strategies. And the Vision 2030 proposed by the Indian Institute of Soil Science, according to the strategy and the framework, and they have to force 5 strategies. One of the strategies is minimizing soil pollution by using sustainable practices. And I thought if the time permits, I would like to explain very briefly about the phytomining or agromining strategy. And after harvesting, and so many benefits, in addition to the ecological benefits, we have to obtain so many socio-economic benefits. We can obtain several biodiesel, biogas, bioplastics, biomass, biopolymer by these biomass, plant biomass. So that's why once we used the acid mine drainage, this is the Dabosan mine site, and we can utilize it for the can-off plant for remediation. And we, in our research group, we utilized the Jatropha carcass and for the phytostabilization approaches in the Dabosan mine site. It's a polymetallic mine site to remove the stabilization technology, and several plants, as I have already mentioned. And Professor M.N.V. Prasad has highlighted several suitable ideal plant species from different mine sites like limestone, coal mine, lignite, and this is the NALCO, National Aluminium Company in Angul site for remediation aspects, and manganese mine site by Niri. They can operate in Niri, and this is a mine reclamation from Asia Pacific region. Grevillea species. First, they have to utilize in the year of 1974. And the Grevillea is a Proteaceae family, is a manganese hyperaccumulator pieces. Phytolacca americana, this is the recent report, and several Prosopis juliflora and Jatropha carcass utilized for mine sites in Lucknow. And this is the transgenic poplar plant, or Salix familiaris, used for copper mining area in Germany. And this is some of the agronomic practices like application of fertilizers and proper water maintenance and other things. Suitable agronomic practices also improve the biomass content of the above ground plant pots and also higher accumulation. And Professor M.N.V. Prasad, in associated with the Thai group people for the mine site reclamation by using the indigenous grass species and to arrest the soil erosion in the open cast mine in Thailand area. And this is the phytomining strategy is nothing but once we can harvest the plants from the plants, above ground plants, once it's accumulated into the above ground plant pots, we can harvest the above ground pots, and we use it to pyrolysis or other methods, and we have to ash, and the ash we can extract the metal ore and other things, our value added products, and that's the strategy was coined by Professor Rufus Chaney from Matsville in Remediation, and this is the laboratory field study. And this is the serpentine soil, ideal soil for phytomining or nickel soil. Ultramorphic, I have already mentioned, and this is different bio-ore processing options. And on the field condition, after harvesting, ash and purify and smelting, finally they utilize it for nickel finished products also. And this is some of the field experiment by nickel extraction from Elisa Murrell in southwest Aragon, and this is Balkan, Albania. And we used improving the phytomining by using the agronomic practices, fertilizers application, and phytomining by Philanthus balcui. And this is the recent report by Philanthus Rufus Chaney. Yes, I have already mentioned father of phytomining, Professor Rufus Chaney. And to honor Professor Rufus Chaney, they have utilized the name of that plant, Philanthus Rufus Chaney, used for nickel phytomining. And Berkia codi, it is one of the why we need to pay more attention about indigenous hyperaccumulator. This is indigenous one, Berkia codi from South Africa. It grew very well in that South Africa. But if we have to transplant it into that New Zealand soil, it didn't grow very well. So the same plant species, same soil type, but the growth condition is not feasible and the idea is not feasible. And so that's why we need to identify more indigenous plants according to that particular site condition. And some other plant species, and due to time constraint, I am not going to in-depth about those things. And even for Rhinorrhea bengalensis, approximately 2.3% of nickel accumulated in that bark or phloem tissues. And so many challenges and research priorities. In this seminar, so many young minds are there. So many research priorities are there. We need to identify the nickel contaminated soils, develop nickel phyto availability assay product procedures depending upon the different metal crops, and the need for increased survey tropical region, breeding conditions. We need to improve the cultivars, breeding suitable cultivars, and greenhouse or growth chamber trial for nickel uptake, and other plant management practices, fertilization, crop rotation, mixed cropping for nickel in yield, harvesting technique, and exploration more methods to identify the high value added products like other salt, nickel salt, and other things. So many things like gold also extracted from that by using plants in different gold mine in Australia by using cyanide heap process. And these are all the process, but the profit is a linear relationship between the cost wise and the soil gold concentration. And this is agromining, and this term was coined by leading Professor Alan Becker and Chu Ranglayan, our former professor, and Rufus Chaney, Alan Becker, and so many leading personalities. They have to coin it on the eve of World Earth Day on April 22nd, 2015. They coined the term agromining, and that metallophyte or hyperaccumulator plants can be utilized for agricultural purposes in some poor developed area, especially DR Congo. And they have to cultivate such kind of the hyperaccumulator plant and get the economic benefits similar to that of the agricultural crops. So they have to utilize such kind of the practices. Cost-wise, it is a feasible one. So this is the Philanthus Rufus Chaney. A new hyperaccumulator was identified in Sabah, Malaysia. And this is the same thing. Nickel agromining practices is going on the field condition. Even from the beginning, wild 6 to 9 months, and after 2 years, the planting is 6 to 9 months after planting, and this is after 2 years of planting. And they have to publish the concentration and agromining metal crops about that thing. And the different species, Noxia in Australia, Austrian serpentine soil also. And these are all the global target region in Mediterranean and Eurasian region, tropical Asia region. Some of the ligneous species and herbaceous species. And the strategy is similar, and we utilize it for remediation of contaminated soil, and we utilize it for agricultural practices in the low productive land. The main thing is final goal is to extract the metal ore or metal finished products. So that's why the phytomining, agromining is the recent strategy, and several ecosystem benefits also be obtained. And 2 large-scale agromining projects, agronickel and life agronomy, is going on in Europe in a network project. Okay, and some of the another important field phytomanagement practice is selenium remediation. And it is our former professor in US Red Rock Ranch, USDA in California, earlier California. And that field is located in Red Rock Ranch near the central California, and they have to contaminate both the one side is contaminated with selenium toxicity and another side selenium deficiency problem, and we need to identify one solution on single strategy to solve these two problems, and we utilized brassica napus or kadugichedi solluvom by for the contaminated sites, and after the growth very well, and we need to harvest that one from the contaminated sites and extract the oil and mix it blended with the diesel and utilize it for the farm practices. And our main goal is for biofuel for public transportation, and due to some limitations still that strategy is going on, but they have to limit the emission atmospheric standard or within the limited of ASTM standards by Professor Gary Banilos has been already proved that one. And after extracting the bio meal cake and bio oil and seed meal cake, and that the remaining seed meal cake has contains selenium product is one of the things. But the weakness is number of polluted sites, identification of polluted sites, or lack of knowledge, or particular soil mapping even is absent in our country. Lack of agrotechnology for polluted soil, lack of stakeholder involvement. And the stakeholder one also most important to invest some of the land, anything land or some other thing. And moratorium against GM crops, and for the transgenic field studies, it's very difficult because a lot of procedures and get a prior approval for GEOC committee. So many things. So strength is we need to acknowledge the explosion in bioremediation, enormous plant diversity, because in our country we have 4 hotspots with within a 2.4% of the land area, and so many unexplored plant species are there. Indigenous plant species, 3 suitable plants according to the type of the contaminant or type of the contaminated lands, and the sustainability concept, omic technologies. So we have to utilize such kind of the strategy, sustainable strategy even for remediation practices, and to utilize it. And nowadays for nano bioremediation, several nano materials can be also utilized, and I am not going to in depth about those things. And these are all some of the relevant publications in our research group. And these are all the what are the chelating aspects, secondary metabolites, how they have to involved in the metal tolerance mechanism by using the 3 species in a suspension cell culture mechanism by Rakesh Minocha in USDA Forest Service Group. And later we have standardized on the method. So far this is the shortest method to identify the monothiol or phytochelating, separation and quantification of glutathione and other monothiols and phytochelatins within the 34.8 minutes. And this we can save more acetonitrile solvent because it's highly toxic and also highly cost-wise it's very high, and we can save a lot of time and also solvent and also so a lot of other things. What are the chelating practices, and we have to publish some of the reviews and recent book chapters and about the things, and it's exclusively what are the molecular approaches we use it for mine site rehabilitation by Bio-Geotechnologies for mine site rehabilitation by Professor Prasad, and myself and my spouse also published in environmental sustainability in a book in by Springer, and it covered for all the sustainable practices. and in our to maintain the soil quality, and the government of Tamil Nadu has implemented 15 for soil health card மண்வள அட்டை அப்படின்னு implement பண்ணி இது பண்ணியிருக்காங்க. But later the government of India has implemented in 2015, and in our university, with the help of our university authorities, we have conducted every year on the eve of World Soil, we have conducted several farmers interaction program rather than the conference or other things. And the international conference on the eve of World Soil Day, International Year of Soils, we have conducted a international conference seminar and also farmer interaction and soil identification, or what are the soil classifications, soil deficiencies, the nutrient profile, with the association with the government of Tamil Nadu Agricultural Department, Salem district. And we have to distribute soil health card by the government of Tamil Nadu Agricultural Department. And in addition to that, we have to conduct exclusive workshop for mine site reclamation in the 2 years before, with the support of TANMAG, Tamil Nadu Magnesite Limited, SRCL, and the Ministry of Environment, Forest and Climate Change. So these are all some of the photographs for mine site visit for young minds to get more knowledge or explore more knowledge in that reclamation aspects. And also we have to conduct it every year for FESA in the title, Frontiers in Ecobiological Sciences and its Applications. And these are all covered with different themes. And last year theme is agriculture, ecosystems and environment. So we have to conduct it every year with different themes. So these are all the sustainable practices, sustainable development goals. They have to propose the 15 development goals by UNDP, and the phytotechnology, it comes under the life on land, and we need to mitigate the soil pollution that will indirectly help to the so many 8 development goals, including no poverty, no hunger, good health and well-being, clean water and sanitation, as I have mentioned in the first 2 slides. So we need to maintain so many sustainable development. To achieve the so many sustainable development goals, we have to maintain that SDG 15 by using sustainable technologies. So I would like to acknowledge in this scenario by USDA, and when I was working as a visiting scientist in NRS by USA, and China Postdoctoral Science Foundation for giving some funds for postdoc research, MOEF and CC, Ministry of Environment, Forest and Climate Change, New Delhi, and DST, CSIR for funding my research projects, various research projects in phytoremediation aspects, TANMAG, SRCL, ICL, Vedanta Group for mine site visits, and Slovak Environment Agency for getting some visiting fellowships or some other collaborative things. So these are all our research collaborators, and thank you very much for your attention. And if you have any clarification, please feel free to ask if time permits. Thank you, madam.
Hello. 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 Hello, sir. There is some question in both Google Meet and YouTube, sir. Shall I ask?
Hello.
Hello. Can you hear me, sir?
Yes, sir.
So there is some question in YouTube as well as Google Meet. Shall I go to?
Okay.
There's some question from Dr. K. Gautam. What type of the tool?
Sir, I'm not able to get clearly.
Hello.
Yeah, yes, sir.
Sir, one question from Dr. K. Gautam. What type of tool used to construct the phylogenetic tree?
What kind of?
Tools used to construct the phylogenetic.
Several bioinformatics tools are used, because I haven't used any bioinformatic tools for phylogenetic tree, and they have to use some other purposes for the classification of trees for hyperaccumulators, different metals. Some bioinformatics tools they have to utilize it for to make the phylogenetic trees.
Thanks, sir. Next question from SK Das. What's the best Bio-Geotechnology for mine site soil rehabilitation?
Sorry.
Hello.
Yes, sir.
Another question from SK Das.
Ah.
What is the best bio-geotechnology for mine site soil rehabilitation?
It's a phytostabilization is the best option. Phytostabilization, or we need to go for phytoextraction. We need to utilize it for the short annual crop, and we have to harvest oftenly, and utilize it for other purpose like phytomining purposes. But so far, in mine sites, phytostabilization and phytoextraction, two strategies has been utilized. And for acid mine drainage, some of the drainage water, they have to stagnate and at the mine site. That's called AMD, otherwise acid mine drainage. And that acid mine drainage water, when they enters into that agricultural field, that will affect the agricultural soils also. So for that AMD purpose, we utilize it for some of the bioremediation tools by using metal tolerant microbes, or some other microbial consortia also used to remediate that acid mine drainage soil, to remove the heavy.
Okay, sir. Thank you, sir. And another question from Punita. Does any microorganism act as an indicator to find the hyperaccumulative plants?
No, we have, so far I have no idea about we have used it as an indicator or not. Because indicator, hyperaccumulator indicator, we can utilize it with several criteria. I have mentioned translocation factor, enrichment factor, and translocation indices. In addition to that, the metal accumulation criteria. For example, cadmium, thallium, selenium 100 mg. For lead 200 mg, so like the zinc 3000 mg, manganese 10,000. So different threshold values already they have to prescribe. In addition to that, we utilized several factors, biological factors, translocation factor, enrichment factor, translocation indices. And these factors also we have to consider based on the metal accumulation, whether and metal translocation ability, based on the metal accumulation ability. And merely the soil microorganisms, based on that soil microorganisms, we can able to indicate whether it is metal hyperaccumulators. So we can do that. And we can do one thing, even though for that hyperaccumulator plants, and you have to collect that soil rhizosphere soils, and you have to identify which microorganism is dominant for that particular mine sites or particular metal contaminated sites. So for based on that, the tolerant bacterial or fungal or microbial community, you can utilize it, that strategy even for some other moderately metal contaminated sites or in lowland areas and some other agricultural soils, by utilize it by that strategy, microbial bioremediation strategy.
Okay, sir. Thank you, sir. And another question from SK Das. What is the potential of in situ nano phytoremediation strategy to remediate heavy metal contaminated soil?
And nano phytoremediation, and so far some of the laboratory they have utilized it for nanotechnology or nano phytoremediation. But another option, nanotoxicology, is one of the another problem. So in depth about the use of nanotechnology or nano-based aspects in remediations.
Okay, sir. And another question from Punita. If there is any indigenous hyperaccumulative plant in Tamil Nadu?
And for that one, so far in our research group, we have to work on different mine sites in the southwestern Tamil Nadu, and we have to identify some of the plants, and that data when is under process. And so that's why I'm not explore the name of the plant. But so far in Indian perspectives, none of the plants has been included in the hyperaccumulator database, global hyperaccumulator database. We have so many papers in hyperaccumulator and other things, but none of the plant species has been designated as hyperaccumulator from Indian perspectives in that global hyperaccumulator database. But so many journals they have to publish it when they say that plant is also hyperaccumulator based on that laboratory studies. But they have a lot of criteria when they fixed before we can identify it or before we can choose that particular plant is a hyperaccumulator. That plant when they can be utilized only able to accumulate that much amount of the metal, especially in that naturally occurred soil or naturally geogenic in nature, not for laboratory artificial experiment. But in our Indian scenario, so many experiments are worked under either pot culture or hydroponics. They have added unrealistic environmental concentrations like 100 ppm of cadmium, and immediately they said more than 560 ppm of cadmium accumulated in the plant above ground. So that's also hyperaccumulator, but that's not real scenario or real hero of hyperaccumulator. Any plant species that can able to accumulate that much amount of the threshold value, and also based on that so many criteria they have to meet out, and different periodical collections we have to do, and then only we have to meet out all the characteristics, then only they have to designate as hyperaccumulator plant. In Tamil Nadu and our Indian scenario, none of the hyperaccumulator plant species have been included in that global database, my hyperaccumulator database.
Okay, sir. Thank you, sir. Another question from Nahendra Raju. Are there any remedies to balance biogeochemical cycle by using technology?
What, sir?
Are there any remedies to balance biogeochemical cycle by using technology?
Balancing biogeochemical cycles, sir.
Yes, sir. Yes, sir. Yes, sir.
And balancing biogeochemical, some of the things are in mine sites are geogenic in nature. So we need not to balancing the biogeochemicals. Only we need to remediate the contaminated sites or remediate the aquatic systems by using remediation. But balancing the geogenic nature, it's very difficult. So many factors are there in natural environment. So we can't balance by using only a single strategy. Only we can maintain that the metal contaminated sites up to the level. For example, cadmium 3 ppm is the international standard. And even for soil quality, another one important point, even in India, a Ministry of Environment or any other Central Pollution Control Board, they haven't fixed the soil quality data so far. Fixed parameter. Even for water quality parameter is there, air quality parameter is there, but no specific soil quality parameter proposed by the Ministry of Environment as well as for the Central Pollution Control Board. So we followed international standards, EPA or European standard. For example, cadmium 3 ppm is the maximum. We need to maintain that level. So that's why it's a same like that. So we have to maintain the contaminated sites, not for to balancing the biogeochemical cycles nature.
Okay, sir. Thank you, sir. And another question from Royal Singh.
Yes.
Does the extraction procedure affect the environmental standards?
Sir,
The extraction process. Does the extraction process affect the environmental standards?
extraction process.
Yes, sir. Yes, sir.
Which extraction process affect the environmental factors? So I'm not able to get the question.
It's a partial question, I think. It's like an incomplete question. It's a general extraction process mentioned பண்ணிருக்காங்க sir.
Okay, general extraction நம்ம அந்த மாதிரி இல்ல. For example, mining sites அப்படிங்கும்போது we need to utilize the phytoextraction strategy and we have to extract heavy metals up to we have to attain the mark of standard level. For example, as I have mentioned in the previous question answer, if, for example, cadmium 3 ppm or manganese 10,000 ppm. So to attain that particular level, we have to utilize it. For aluminium 1000 mg or 100 mg, 1000 mg per kg. So up to that level in that soil, we have to maintain or we have to continuously utilize that strategy. Even though once we have to extract that above ground plant parts, we use the phytomining strategy. In Indian scenario, So far, that strategy has not implemented. Only in European country and some of the Middle East countries, they have to, not Middle East, in the Balkan area and other countries, they have to utilize that strategy so far exclusively in nickel only. Because the soil, land condition as a site condition or substrate condition is also one of the most important. So they utilize it by hyperaccumulator plants for that particular substrate, like serpentine soils. Serpentine soil is nickel enriched soils. So they utilize it to maintain the nickel concentrations in that particular soil, also remediation purpose. And also they have to extract the nickel ore from that particular above ground biomass by using different processes, and finally they get the value added products, and in addition to that, the nickel ore.
Okay, sir. Thank you, sir. And sir, last question from Akshaya Vijay. Sir, can you hear me?
Sir?
Last question from Akshaya.
Yes, please, sir. Please.
Vijay. Some plants that were grown in highly polluted region, especially plastics. What do you think about, sir?
Some plants can grow, sir. Some plants?
Some plants that were grown in highly polluted region, especially plastics.
Plastics?
Yes, sir. Yes, sir. What do you think about it? And can
I don't know the plastics eating plant. I have no idea about that. Some plants can able to grow in highly contaminated sites, but plastic sites, I don't know. I have no idea about those things. How they have to mention about the plastic sites. But nowadays, even the soil also, microplastics also influence in marine environment, even also marine sediments. And nowadays, they said microplastics accumulated in that soil also. But plastics means normal carry bag and other things. And I don't know in which aspects they have tried. அவங்க சொல்றது சும்மா அந்த கேரி பேக்ஸ் மற்ற பிளாஸ்டிக்ஸ் இருக்கிற ஏரியால அது பறந்து போய் பிளான்ட் பக்கத்துல விழுகிறதுன்னு நினைக்கிறேன். நேச்சுரலா பிளான்ட் பிளாஸ்டிக் இருக்கிற இடத்துல போய் யாரும் பிளான்ட் வைக்க மாட்டாங்க. அதாவது மைக்ரோ பிளாஸ்டிக்ஸ் என்னுடைய இதுல இருக்கலாம், வேணாம் சைட்ஸ்ல இல்ல கண்டாமினேட்டட் சைட்ஸ்ல மைக்ரோ பிளாஸ்டிக்ஸ் இருக்கலாம். அந்த மாதிரி ரிப்போர்ட்ஸ் மைக்ரோ பிளாஸ்டிக்ஸ் அக்குமுலேஷன் இன் சாயில்னு ரீசன்ட் ரிப்போர்ட் இருக்கு 2020. பிளாஸ்டிக்ஸ் இருக்கிற இடத்துல பிளான்ட் வளருதுங்கிறது ஐ ஹேவ் நோ ஐடியா.
Okay, sir. Thank you, sir. Thank you for your passionate answering for the question, sir.
Okay,
thank you.
Thank you very much, sir.
I hand over the session to the Miss Sophia, ma'am.
Yes.
Over to you, ma'am.
Thank you, sir.
Thank you, sir. Thank you. Thanks a lot. Over to you, ma'am.
Okay, sir. Thank you, sir. Thank you for your wonderful talk on phytoremediation, sir.
Okay. Thank you very much, madam. Thank you. And once again, I am much thankful to the principal, head, and other faculty members for giving me an opportunity to share my views in that esteemed platform in your esteem. Thank you. Thank you very much.
Thank you, sir. Good evening, all. I deem it a great honor and privilege to propose the vote of thanks for this webinar. Let me start by giving glory to the Almighty for making this webinar a great success. My heartfelt thanks to the management and our beloved principal, Sir Dr. S. Madhivanan, sir, who gave constant encouragement and support to conduct this national webinar. I wish to convey my special gratitude to our resource person, Dr. Thangavel, sir, for his enlightful talk on phytoremediation. Thank you, sir.
Thank you.
Next, I would like to express my sincere thanks to all the participants for their cooperation for this webinar. I wish to express my gratitude to our head, Department of Microbiology, Mr. D. Kannan, sir, for his support and guidance. I also wish to express my deep sense of gratitude to faculty members of Computer Science Department, Mr. Saravanan, sir, Dr. Benjamin, sir, and lab technician, Mr. Mariselvam, sir, for their technical support in this webinar. My colleague, and not department. Thank you all. Kindly fill the feedback and you'll get your certificates within 2 days. Sir, thank you, sir.
Thank you, madam. Thank you. Thank you.
Thank you, sir.
நல்ல ஃபீட்பேக் நல்லா தான் கொடுத்துருக்கீங்க.